This video lecture is over Chapter 14, Translation and Proteins. Today's topics, we're going to be talking about translation, the process. So we'll talk about initiation, elongation, and termination. And then we'll talk a little bit about proteins, the amino acid structure and protein structure. So in our central dogma of molecular biology, translation is the process of converting the genetic language that's based on the three-letter words that was transcribed into messenger RNA and translating that into amino acids. So transcription takes place in the nucleus. That's where we make our mRNAs, and then the mRNA travels out of the nuclear envelope into the cytoplasm, and then it binds to a ribosome and we initiate protein synthesis. So the players involved in this process are the messenger RNA, that's the recipe. Okay, our ribosomes, that's the protein factory. You can kind of think about that as the chef. The transfer RNA, that's the the mailman that delivers the amino acids, or you can think about it as like the sous chef that delivers the ingredients. And the amino acids, those are our nitrogen carbon compounds. They are the ingredients by which we will make our proteins. So just to clarify where things are moving, our mRNA, once it moves into the cytoplasm, our ribosomes are going to recognize the mRNA and attach to the 5' methylguanosine cap. At that time, the ribosome will then start scanning the mRNA and searching for the start codon. When it finds that AUG, that methionine start codon, then tRNAs will start to supply the amino acids in the order specified by the mRNA reading frame. A ribosome will then assemble the polypeptides in the order that the mRNA dictates, and it'll continue until the ribosome reaches a stop codon or a nonsense codon, at which time the polypeptide chain will be released. So our two accessory players besides the mRNA are going to be our transfer RNAs, which bring the amino acids, and our rRNAs, or our ribosomes. This right here is a transfer RNA. Okay, it's a simplified version drawn in a 2D format. We call this a clover leaf. And there's a few things you'll see. Okay, on the 3' end, we have what's known as the acceptor arm. This is where the amino acid will attach to the tRNA. Okay, we also have what's known as an anticodon arm. This section right here will bind to the mRNA. Okay, and on that mRNA where it binds, it will then be the complement that we find for of the mRNA, and it's what designate or what stipulates which amino acid gets attached. It's complementary to one mRNA codon. We talked last time about the wobble hypothesis. So you see how this function, how we have this clover leaf where it's landed. We're going to start making hydrogen bonds between these nucleotides and those on the ribonucleosides that are found on the mRNA. And because it's not flat, you can see how these hydrogen bonds are going to kind of get bent, and there's a little bit of a wobble space. On top of that, we have this ionosine that's found on a lot of our tRNAs that can bind to just about any ribonucleoside. And that is what allows for that third position wobble, right? Both the shape and that ionosine. Okay, so I told you this is just the secondary structure. If you look at the tertiary structure, you start to see how this is much more complex and how it kind of folds up on itself. Again, if we look at that anticodon, we see that ionosine. This particular codon would be complementary sequence of alanine, and it could bind to any one of these codons. So you see on the 5' end of our mRNA, in this third position, that's what's going to bind with this ionosine, and that's what gives that degenerate nature to our code, and that wobble would allow this position to be a little bit more flexible, whereas these other first two positions and most of our codons are very, very rigid. tRNAs go through a process called charging. This is the process by which we attach the amino acid. This is occurred by the use of an enzyme called amino acyl tRNA synthetase, as well as the amino acid that's designated by the anticodon and ATP. So the enzyme recognizes the anticodon on the tRNA, and that specifies its specific amino acid. We create this activated enzyme complex. So in this point, you'll see we take the ATP, we put it in there, we get an amino acyl adenylylic acid. It then works and converts this and charges the tRNA using this amino acid and the ATP in the enzyme. It'll attach it to the acceptor arm. Okay, and that makes it activated. This tRNA can then bind to the ribosome and be utilized in translation. Now our rRNAs that are involved in this function make up our ribosomes. Ribosomes are the workbench of the cell. Okay, they are composed of two subunits. We have what's called the small subunit and the large subunit. And both of these subunits reside in the cytoplasm. They can be either united or separate. And when translation is initiated, they will become united together if they are separate. And unlike how tRNA has a specificity for only a single amino acid, our RNAs can bind to any mRNA. Okay, there's a couple spots on here we should be familiar with. So we have the A slot, which is short for the acceptor site. This is where the tRNA will first enter into the ribosome. We have the P site, or the peptidyl site. This is the site in which the polypeptide chain is elongated. And then we have the exit slot. This is where the tRNA will leave once it has donated its amino acid. Now, prokaryotes and eukaryotes have slightly different rRNAs or ribosomes. In prokaryotes, two subunits are designated by their size. So we have the 23S RNA, and that makes up the large subunit. The small subunit is made up by the 16S. In eukaryotes, the gene responsible for the small subunit is 18S, and the large subunit is the 28S. All right, now we've gone through all of our agents. Let's look at the ingredients, what we need. So we have a ribosome, we have our mRNA. We're also going to need initiation factors to help us start translation. We're going to need elongation factors to help us with moving the mRNA through and attaching our tRNAs onto it. And we're going to need our initiator tRNA. So this is the tRNA that binds to the start codon, and it has the amino acid methionine. Okay, again, our start codon is AUG. Initiation begins when initiation factors bind to our small subunit and help that subunit bind to the 5' 7-methylguanosine cap of our mRNA. Okay, then the small subunit is going to start to scan or search our mRNA looking for our start codon so that it gets the correct reading frame. That start codon is going to be aligned in the P site. So it gets aligned into the peptidyl site. Okay, and our tRNA that's going to carry the 7-methylguanosine, the tRNA that's going to carry the formal methionine will bind to the mRNA in the peptidyl site. The P site is filled with our tRNA. Then the large subunit will bind to the tRNA, and initiation factors will then be released, and we'll have our completed ribosome, both the small subunit and large subunit together, with the start codon aligned in the peptidyl site, our tRNA bound in the peptidyl site, and our next amino acid is going to be moved into place by the appropriate tRNA. This moves us into now out of initiation and into elongation. So elongation, the codon following that start codon is now found in the acceptor site, so in the A site. And it's going to specify the next charged tRNA that can enter. So we're in here, elongation factors are going to then bind to the tRNA and they're going to facilitate its movement into the acceptor site. Once it's been moved into the acceptor site, the anticodon arm is now in that A site, and there's going to be enzymes that bond the amino acids attached to the acceptor arms in the P site to the A site. So what you're going to see is the one that's actually in the peptidyl site is going to be bound on to the amino acid in the A site as we shift. So the codon is going to shift, and the tRNA that was in the peptidyl site will move into the exit slot, and the one that's in the A slot is going to move over into the peptidyl slot, and you'll see that we now have a tRNA that has, in this case, a phenylalanine and a methionine attached to it. And so we elongate this way. This all happens at the same time. So tRNA enters into the A site, and as they shift together, we move the amino acid onto this tRNA, which then resides in the peptidyl site. And that process is called translocation. So we move from one codon to the next, and this is what just keeps happening. The mRNA moves through with this. It's not showing that these are bound, but they really are bound in this position. A site is now open. The next tRNA is moved in by the elongation factors, and our polypeptide chain continues to grow. And elongation is going to continue until a stop codon is reached and is moved into the A site. Now the stop codon, there is no tRNA that can enter the A site for the stop codon. Why? Because there's no amino acid for this. This is a nonsense codon. So when nothing enters in, that tells the ribosome that we have completed our polypeptide. So the tRNA in the P site has a complete polypeptide chain, and then we have what are called release factors that come and break the final bond on our T site, and it releases this polypeptide. So we break this bond right here, and the polypeptide is released out of the ribosome. The final tRNA moves into the exit slot. The ribosome disassociates itself from each other, and it's ready to start initiation all over again with another mRNA. Okay, something I should also mention is that the tRNAs, as they get released from the exit slot, they can be recharged. They're recycled. Okay, and the mRNA in theory could be recycled as well until it's degraded. So here we can actually see in these slides ribosomes, the mRNA, being translocated through. We can see the polypeptide chains in this one forming off the ribosomes. And our polypeptides, they build our proteins. So our proteins are the building blocks of life, right? Following translation, our polypeptides are then going to begin to fold and assume higher orders of structure. This form equals their function, and the structure dictates what function they can have. That three-dimensional conformation, that's really essential to its protein-specific function. All proteins are made up of amino acids. Amino acids have the same type of structure. There's a central carbon, and attached to that, we have an amino group, okay, that has a positive charge. We have a carboxyl group that has a negative charge. And then we have a radical group. And this radical group is what makes are amino acids specific. Now, you do not need to know all the amino acids for this class, but there are 4 kinds of amino acid radical groups. Okay, the first group are what we call our hydrophilic. These are the ones that like waters. Okay, so hydrophilics right here. Hydrophobics are our other group. Then we have our positively charged or basic or polar radical groups. And then we have our negatively charged or acidic polar groups. And these amino acids, they are bound to each other by a peptide bond through a dehydration reaction that forms between the amino group and the carboxyl group of adjacent amino acids. As far as structure of a protein goes, there's 4 structure levels. Okay, the primary structure is just the polypeptide chain order. Okay, secondary is involved in spirals and sheets. Tertiary is now the bending, and the quaternary is where we bring in different motifs groups. And these 3-dimensional conformations are what gives that protein its function. Okay, how it bends and folds on itself is going to dictate what it can do. And what dictates how it folds and bends is the primary structure. So again, primary structure is just the sequence. We can look at the secondary. The 2 basic types of secondary structure we have are either our alpha helices or our beta sheets, and how they kind of form together and create this. Then the tertiary structure is how our radical groups then follow. So we start having different types of motifs like zinc fingers, other stuff. And finally, our quaternary structure is when we have multiple polypeptide chains then associate with each other. Okay, it's that tertiary structure really that is directly related to its function in protein. Now, how proteins get folded, there's molecules called chaperones that help fold the tertiary structure of a protein. They mold that protein and push it into, basically pushing radical groups closer together so they can form the chemical bonds that they need to do. When we get to that quaternary structure, that's where we have 2 or more polypeptide chains in a single protein that kind of fold together. And we do have some post-translational modification. So some proteins can be modified after they've been synthesized, but we're not going to really spend a lot of time on that. You can take cell biology, and we'll talk a lot more about that in cell biology. That, in a nutshell, is translation and proteins.